Okay, so welcome to lesson number 7 today. Last time we discussed in relative depth modeling in neuroscience, theoretical neuroscience, computational neuroscience, in particular single cell computation. And today I want to go into the Blue Brain project, the Human Brain project. I call this lesson number 7 cortical networks, which we shall focus on cortical networks. We discussed the cortex before, but in particular, I want to call it Out of the Blue project, which is, of course, a game between blue and out of the blue. And we shall try to understand what do we do, what is the aim, what is the purpose of this project. So I will start with a description of several mega projects. So the Blue Brain project is only part of several of them that are now ongoing, just to highlight a few of them. Then I will discuss the cortex very briefly, just to give you an introduction to the cortex, and in particular to the cortical column, the notion of a column in the cortex. And then I will show you that the column, the cortical column, is used or was used, now we are going further, but was used as a test case, as a proof of principle, whereby we want to see whether we can take whatever we know about this column of the cortex, can we simulate aspects of it? And then eventually I want to describe the Human Brain project, which is much beyond the Blue Brain project. And I would like very much to be able to emphasize the difference between the Blue Brain project, which was the test case, and the Human Brain project, which we just started. So I promised you a perspective on several large mega projects. When I say mega, I mean this is really a lot of investment, huge summations, billion of dollars, billion of euros. So I chose just 4. I could choose a little bit more. So one of them is the Allen Institute in Seattle. A very interesting place, a very interesting concept by Paul Allen, who developed this institute, which is growing very nicely, very fast. They already, in a sense, completed the first mission, which became very useful for neuroscience, what they call the mouse or the human brain atlas, whereby they develop methods and also analyzed the expression of genes, different genes in the brain of the mouse, and now they are doing the same on the human brain, which is, as I said, a very important endeavor, because we would like to know what genes are being expressed in the mouse, in the healthy mouse brain or in the sick mouse brain, whereby hopefully we can better understand the interaction between certain diseases and certain genes. But now recently they started a new project there in Allen Institute focusing on the mouse visual system. And one may wonder why the mouse visual system. The mouse is not the best seeing animal, but still they decided, after a big debate on what should be the next project, they decided on the mouse visual system. And the reason, part of the reason, is that we know a lot about the mouse visual system, both anatomically, physiologically, and also one can develop many new tools, genetical, optical tools, to study the mouse visual system or any other system of the mouse. And so the mouse is a very good experimental system for understanding a whole system, in this case the mouse visual system. So Allen Institute in Seattle is a very prominent, very promising, very successful institute for brain research. The next one is Janelia Farm, not far from Washington, D.C. And they're supported by the Howard Hughes Medical Institute. They develop really an industrial scale new institute, very impressive also architecturally. And there they develop methods and also very fascinating experiments to study the interaction between anatomy of particular systems, mouse, C. elegans, and other lower level, so to speak, animals, to connect anatomy and physiology to specific behavior of subsystems. And this is really extremely modernistic, I should say futuristic center, both technologically, also aesthetically, and also conceptually, how to go about studying particular systems. Very expensive, very successful, very new. Highly recommended to visit there just to see how modern science is being performed. I will talk a lot today, of course, about the Human Brain Project because I'm involved in it, and it came out from the seed of the blue brain. I just want to now just say that it is located as a center at the EPFL. It's a whole European project, but the focus, the center is at the EPFL in Lausanne, in Switzerland. The idea is to develop new platform, not new experiments, but new platform to study the brain, new way to integrate data, to integrate knowledge from different disciplines, both from physiology and anatomy and computation and engineering and other technologies I will mention later on, and hoping that this integration of data under one umbrella, under one platform, will enable to really enhance, accelerate our understanding of the brain, both in disease and in health. And eventually, the hope is, of course, to try to develop new treatments from brain diseases due to this encompassing, this integration, new platform that we propose. And recently, I already mentioned it in the first talk. I'll mention it a little bit more today, although I'm not going to discuss it, of course, and it's only starting, so it's ongoing debate. What exactly is the President Obama Brain Activity Map, or BAM, Brain Activity Map project or initiative? But the general idea is to develop new techniques to measure the activity at the single cell level, the spiking activity at the single cell level from millions or maybe even billions neurons simultaneously in the living, behaving brain. So that's the big mission of the Brain Activity Map of President Obama, which is very debatable. As all such big projects are always debatable, I will mention later on, but you already studied at depth the Hodgkin-Huxley model and what spikes mean, the spike mean. And so the Brain Activity Map is to record many spikes in the behaving brain, hoping that when you see a brain active at the level of single cells, you may be able to understand certain aspects of coding, decoding information, representing information, and also what happens in the diseased brain. So that's the idea behind the concept of recording from many cells. We cannot do it today in the behaving cells at the level of millions or billions. We can record from hundreds of cells or maybe thousands of cells, but certainly not at this level, and that's the mission of the BAM project. Okay, but this was just a survey, and I already mentioned diseases, and you may know that one of the aspects of brain research, and not the focus of all brain research, is the aspect of diseases in the brain. Unfortunately, and I mentioned it in the first talk, unfortunately, this very delicate, very fragile system generates diseases of many, many types. Here all the names of known neurological or neuropsychiatric diseases under a project called the Desisome, an attempt to systematically, based on what we know about genes of certain diseases, of Parkinson and Alzheimer's and schizophrenia and depression and all these many diseases that we know today, what is the common ground, what is the foundation, genetic foundation, and other correlations between different diseases. So this is an attempt to systematically somehow put all the diseases under one framework. And this is also part of a big attempt worldwide to make sense, so to speak, of diseases. This is also part of the Human Brain Project, which I will mention later on. So diseases, of course, is a big drive for all these big projects. Humanity needs to solve the problem of the brain. I mentioned some solutions that are more symptomatic. We would like to understand the depth, the deeper the aspects of diseases in general. So we know all these diseases emerge typically at older age, but not always at older age. For example, autism is not a disease of an older age, but Parkinson maybe yes, and Alzheimer's certainly. So we need to understand all diseases, and in particular, and that's a point that I want to emphasize, and we already did, that all these diseases, all brain diseases, emerge from some aspect of activity or anatomy or both, some misbehaving region in some sense, or a connection, or the excitability, the spiking activity, or both in a particular set of networks. Something goes wrong in many, maybe locations. Together, this system generates a behavior, an output that is not the appropriate one, and disease comes up. Whatever disease means, there are definitions of disease and so forth. And I already mentioned before, I think in the first talk, I already mentioned the work of Professor Hagai Bergman from our institute, from the Hebrew University, who is working on a particular region of the brain. I already showed you an activity of a, so to speak, normal, non-Parkinsonian piece of a brain. In this case, it's not the cortex, recorded not from the cortex, but from deeper brain regions, the basal ganglia. But the principle holds for any region. You see an activity that you call normal, that is not associated with Parkinson in this case, and then you see an activity, electrical activity, spiking activity, and now you know that each one of these blips, each one of these signal is called a spike. So you can see a spiking activity of 3 cells in this case recorded simultaneously, whereby you see that the change in activity is associated with a disease. So we want to understand what is the mechanism, what could be the mechanism in this particular network that generates a wrong electrical activity underlying Parkinson. So that's one of the big missions of many of these projects, and including the Human Brain Project. Not only describing the disease electrically, but understanding the underlying, the underpinning mechanisms, synaptic, electrical activity, combination of them, and so forth, that gives rise to this and not to this. Hoping that by understanding this, we will better understand the disease.